someone recognizes their phone, then you can come get it here. Otherwise, I'm just going to put it here for now, but so you know. Sorry?
One person will have the real aha moment.
Yes, exactly. Okay, so moving on to our next talk, our next master talk and fourth talk is going to be by Gabrielle Michon, and she's a master student of molecular and cellular neurosciences in Paris, if I'm correct. And that sounds really interesting, to be honest. And she's also a member of ALPS since recently, where she's mainly taking care of the ALPS newsletter. And besides her studies, she's working at the Paris Brain Institute, where she's also conducting the research that she's going to present. And she's going to present research about mice's behavior and neural activity during various doses of LSD. And besides that, she came all the way from Paris to give this talk. So please give her an extra warm welcome to Gabrielle Michon as she comes on stage.
Thank you, Dave, for this introduction, and thank you everyone for being here. So my talk will change a bit from what you heard previously, because we were talking about mice. So this is very different. As Dave said, I'm a master's student working at the Brain Institute in Paris, and I'm supervised by the professor Eric Berger and by Daniela Dominguez, which is doing her doctorate. So first, I will give a whole introduction of what we know on psychedelics, and especially about LSD in animal research, but also in humans. Because I know you know most about human, but let's go a bit in animal research, please. So first, as early as the 16th, psychedelics appears as promising in the treatment of various psychiatric disorders, such as alcoholism, fascinating psychiatrists, scientists, and artists. However, due to their recreational use, psychedelics were rapidly prohibited and classified in the Schedule I drug in the Controlled Substance Act, leading to a freeze of research for decades. However, we are now going to a resurgence of the psychedelic research. So what are psychedelics? So as we mentioned, psychedelics were defined by Humphrey Osmond, and it comes from the term psyche, which means soul, and delos, which means reveal. So basically, psychedelics makes the ability to reveal the mind. And indeed, psychedelics were shown to induce ego dissolution, alternation of perceptions, and hallucinations. Thus, they are always referred as hallucinogens. But today, I will only talk about classical psychedelics, which are all at least partial agonists of the 5-HT2A receptors. And among all those psychedelics, LSD is the one with the most complex polypharmacology. However, it is the one that is also the most understudied, and it is notably due to its bad reputation. But we can wonder if this reputation is fair. And indeed, early attempts to use hallucinogens in therapeutic was conducted in Canada by Offer for the treatment of alcoholism patients. And it was found that nearly half of the patients stopped drinking after being exposed to LSD. And since then, clinical studies demonstrated that LSD significantly and rapidly decreased anxiety and depressive symptoms. In these graphs, it is in patients with terminal cancer. But also, they decreased OCD symptoms. And really interestingly, this was done in animals. So we can see that indeed, psychedelics were found to decrease anxiety, depression, and PTSD-like behavior in rodents. But so now we know that psychedelics work in a way, but we don't know how. So for now, I will explain a bit what we know about LSD and animal research. So first, at the molecular level, We know that LSD is a particularly potent agonist for the 5-HT2A receptors. And indeed, serotonergic receptors, and especially the cortical neuronal pyramidal 5-HT2A receptors, were found to be really important for the behavioral effects of hallucinogenic compounds. And this was assessed notably with antagonist studies that were done here in vivo in animals, but also in humans. And this has also been assessed with KO studies. So a KO is like you're just deleting the gene in animal. So in the graph in the middle, the mouse do not have any 5-HT2A receptors. But there are some differences between 5-HT2A agonists. Some are hallucinogenic and some are not. And we can wonder why. And one of the reasons why is that LSD has high affinity for many other receptors. Indeed, it has high affinity for all the serotonergic receptors, but also dopaminergic, histaminergic, adrenergic, and many others, certainly. And here is just like an example, but I know that there are a lot of missing here. It's just to show you that it is a mess. Then, another reason why there can be these differences between hallucinogenic and non-hallucinogenic 5-HT2A agonists can reside in the pathways. So indeed, we know that the 5-HT2A receptors is a GQ protein-coupled receptors, and this activation will increase the production of inositol triphosphate by the recruitment of phospholipase C. However, it was recently demonstrated that indeed LSD change the conformation of the receptors and promote the recruitment of another signaling pathway, the beta-arrestin 2 pathways. And this was also shown to be related to the reopening of the social learning period in mice. So now that we talked also a bit about plasticity, yes, we know that psychedelics affect plasticity in a good way. Indeed, LSD were found to promote neurogenesis and to promote the increase of the density of spines. And also it was found that there were an increase of the immature spines as opposed to the mature spines. This was done first in vitro. Then we wanted to see it in vivo, so in mice. And again, we saw that LSD increased spines density in mice. And this was functional, as we can see here. So then, psychedelics induce structural and functional neuroplasticity. And then you can see on the last graph that ketanserin, which is an antagonist of the 5-HT2A receptors, meaning that it blocks the action of other agonists, were found to block this increase in spine density. So neuroplasticity is in part mediated by the 5-HT2A receptors. But here I'm just simplifying things, because again, there are many other components. So now another reason why psychedelics can function, it's because they dampen neuroinflammation. And indeed, it has been proven that when putting psychedelics in vitro with microglial cells, after being exposed to a pro-inflammatory agent, it decreased the pro-inflammatory production of TNF-alpha. But then, when you put also LSD or other psychedelics such as psilocybins with microglial cells which have been exposed to pro-inflammatory agents, you also see that it changes their phenotype. So normally when microglia are pro-inflammatory, they have a certain type of phenotype, and then you can assess if psychedelics decrease these pro-inflammatory states, just looking at the circularity, for example. So this has been done in vitro. But then in vivo. So these are models of mice of PTSD. And we saw that so psychedelics decreased the production of ROS, which is a pro-inflammatory markers in the prefrontal cortex and also in the hippocampus. So the neuroinflammation hypothesis can be also a reason why psychedelics function and have these therapeutic effects. But we still don't know. So the main question, and how psychedelic works. Then we can wonder how they works at the neural level. So in humans, neural activity was highly measured with fMRI. And in fMRI, it was found that LSD notably altered the resting state functional connectivity. Namely, it increased the within network, the between network functional resting state, the resting state functional connectivity, but it decreased the within network resting state functional connectivity. These results were found to be a bit inconsistent. There are a lot of different findings, and I'm not an expert on fMRI, so I will not spend so much time on it. But there are many theories of why it can also function and how it functions. And then we can wonder how LSD works at the neural level, but in mice. So in mice, there are 2 recent studies that have been published in which they mapped the whole brain activity evoked by psilocybin and ketamine using the newly developed technique iDisco. So iDisco is a technique that I use also in the lab to make the whole brain transparent. And you can also put it in with immunolabeling, and so then you can immunolabel CFOS expression, for example. So CFOS is an immediate early genes that has been found to be a proxy for neural activity. And in their studies, they found that psychedelics commonly increased cortical but decreased subcortical neural activities. However, this studies has been done with psilocybin and ketamine, but not with LSD. And as we mentioned, polypharmacology of LSD is much more complicated. So it is still unanswered. So now we can wonder how LSD work at the behavioral level. So in mice, in humans, sorry, this has been done using subjective questionnaires. And it was found that psychedelics provoke potentially harmful effects such as ego dissolutions or hallucinations, but also so-called good drug effects such as increase in insightfulness, blissfulness state, feeling positive mood. And these mystical experiences were also correlated with the therapeutic effects. However, this has been found to be really not inconsistent, I will say, but really dependent on the individuals, but also on the setting. And notably, there is a study in humans from Griffin in 2006 in which they found that even 80% of people had an improvement in well-being, 30% of those same people had a really increased and strong anxiety and fear. Thus, now we know that psychedelics have really different effects on different people, and we need more data for that. And because psychedelics are highly dependent on the context and on psychological setting, it is also cool to use animals because we can really control the environment. So now, what does LSD do on behavior? You might hold that wonder this question. There is a behavior that is called the head-twitch response that is really important. So this behavior is a really rapid side-to-side movement of the head. So basically, the mice is doing like that many times. And this behavior was found a proxy for hallucinogenic potential of a compound. It is a really consistent behavior that was found to be induced by hallucinogenic 5-HT2A agonists, but not by non-hallucinogenic 5-HT2A agonists. For example, lizuride does not induce the head-twitch response. Then the head-twitch response were found dose-dependent. This was found to be, these studies were done with other psychedelics, but it has never been done with LSD. So now, I want you to see the head-twitch response. It was described in the literature as a really easy to capture behavior, but believe me, it is not always the case. So, are you ready? So please concentrate on the head of the mice. I know the video quality is not that good. Up. Did you see it? Who saw it? Okay, you're sure. Second time, with a bit in slow motion, because I know it's very difficult to see. Now you will see better. Okay, so please look at the head. Did you see it? Who saw it? Please raise your hand. Okay, much more people. And last time, but frame by frame, so that everyone can really capture the movement. This is it. You see? Really fast, really difficult to capture. And I had to look 100 videos like this of 1 hour. Really cool moments. So now this is not only the only behavior that LSD does to mice. We can see really cool things. So sometimes the mice is just licking the walls. Sometimes he's just like scratching like this. And also there is a natural behavior that the mice is doing when it wants to explore its environment. The mice is rearing. So the mice is just like on her back pose, hands like this, and looking around. And believe me, with psychedelics, it's rearing a lot. So he's just here in the middle of the box. Cool, right? So now that you know that, I can go to the literature and my projects. So even if all what I described to you before is a big advancement in the field, there are a lot of things that we don't know yet. And we mentioned, for example, the effects of LSD on a long-term scale, because we said that indeed LSD could have a potentially harmful effect for the heart, because indeed it's activating the 5-HT2B receptors. And we also don't know how LSD doses can influence the response of someone, and also what it does to the brain. And this is really important, notably to do precision medicines, which aims at giving the right dose at the right person, but also at simply the right medication. Because we know that LSD is different from psilocybin, from DMT. All those compounds are really different at the end. So now the research that we are doing in the lab. So we want to characterize the acute effect of a large spectrum of LSD doses on mice. Mice behavior. So namely the locomotion metrics, the head-twitch response, but also anxiety-like behavior. And then we want to map the acute effect of a large spectrum of LSD doses on the whole brain neural activity in mice. And a last objective was to determine a moderate dose in mice, to then do another experiment in a mice that is a model of OCD. So first, because I know you do not know anything about animal research, I wanted to show you a bit how we can study the effect of LSD in animal research. So the first test is the pre-pulse inhibition test, and this test is used to see the ability of the subject to filter a sensory information. So basically the mice is just put in a box, we give a pulse, a stimulus, and then we measure the startle response. So the startle response is just a reflex of the mice because it is afraid, and so it's doing like this. And when you're doing a pre-pulse, before the pulse, then you can see that the startle response decreases. But with LSD and other psychedelics compounds, this response does not decrease. And this is notably also because we know that psychedelics affect the filtering of sensory information. Then, another way to assess the effect of psychedelics on behavior is the drug discrimination test. So basically, the mice is put in a box with 2 levers, and it's injected with the drug. The mice learn how to recognize the drug, how to recognize what the drug does to his body. And then you give a drug randomly, and if the mice recognize it, it presses the lever. Really intelligent, really. So now the 2 other tests are the most used to study behavior. And indeed, it is because with those tests, we can really capture unconditioned locomotor activity and exploratory behavior in mice. And also, those tests are really cool because they're absolutely not invasive. So the mice is put in the box, and he's just walking, doing his life. And then you video capture. Pretty cool. And you can see a lot of things. So that's nice. No suffering, that's nice. So, what did we do? So first, we used only male mice in our experiments. They were 3 months old because 3 months for mice is an adult mice. They were put in an inverted cycle for 1 week before because we as humans want to work during the day, but mice, as nocturnal animal, want to walk during the night. So we just reverse the cycle so we can observe them in the night. Then we handle them for 2 weeks. Handling means going and see the mice. They can smell us, they can go over our arms, so that when we will do the experiment, they are less stressed. Then the day before the experiment, we put the mice in the box. So like this, is go around, it see how it is, and during the experiment, it will be less stressed. So then for the experiment, we put the mice for 30 minutes in the box for video recording for baseline. Then we injected the mice 1 of the 7 doses, including saline, that we had of LSD. So as you see, this is really a large spectrum of doses. And the mice were put again in the box for 1 hour. Then we sacrificed the mice and we analyzed the data. Yes, sorry. I know this is the not cool part. So for data analysis, we notably used a software Bonsai, which allowed to track the position of the mice. And only with the position tracking, you can have a lot of information. And we also recorded the head-twitch response. So I was the one just like scoring the head-twitch response. So looking all the video and say, okay, here there is one, there is one. Really cool work. So the first result that we have, we at first wanted to assess if LSD has an effect of the anxiety-like behavior of the mice. And we can see that with how the mice is going in the open field. So normally the mice has the ability to avoid center area because normally they can be predators in center field and in bright field. So they tend to go in the corner or in the walls. And so here, what you can see is the occupancy place of the mice in the open box. So these data are captured from the drug phase and they are normalized by the baseline. So when you see red dots, it means that the animals spend more time in this position compared to the baseline. And the blue one, it means that the animals spend less time in these places than in the baseline. So overall, we can see that the mice were a bit more stressed than compared to the baseline, because it seems that the mice is going a bit more into corners. But I think this is pretty normal, because the mice was injected. So you cannot expect the mice to not be stressed at all. But what is really interesting here is that if you look at the corners with the increasing doses of LSD, you can see that they are starting to be a bit larger. So the mice is going to explore a bit, to go away a bit from her corner with increasing doses of LSD. Which means that probably with increasing doses of LSD, it is a bit less stressed. Then sorry, I forgot to change. It's not occupancy, it's immobility. But another way to assess for the anxiety-like behavior of the mice is to quantify the mobility. So in these plots, we calculated the percentage of time the mice were immobile every 5 minutes, normalized with the baselines. So again, overall, we can see that the mice were a bit more immobile, meaning that they were a bit more stressed. However, what is really interesting is that you really have this dose dependency effect. You can see that the yellow line is 300 doses. The immobility is pretty low, but with 0, the immobility is pretty high. So with increasing doses of LSD, you have a decreased time of immobility, which goes in the right way for us, because it seems that LSD seems to decrease anxiety in mice. Then we wanted to assess for the locomotor activity of the mice. And similarly, we measure the locomotor activity every 5 minutes compared to the baselines. And again, what is really interesting to see is that we really have a dose response following an inverted U-shaped curve, with the peak being around 300 micrograms per kilos. And this has been well reported in the literature with other substances, but not with LSD. So this is also really cool that we have the same. This is not really important, so I will go through it. So now I'll talk about the rearing. So this is really cool to see. And you know, as I told you, scoring videos manually, it's really boring. So what we can do is just to do it automatically, machine learning. So how we do it in mice, we just have to label the mice with different points, saying that this is nose, this is here, this is tail. And then we train a software multiple times, so that then the software is able to recognize different parts of the mice. And then you put the skeleton in another software, which is Behaviour. And Behaviour allows to recognize the dynamic of the movement. And like this, you can identify many behaviors and quantify them easily. I will say, yeah, pretty, because it takes really a long time also to optimize the software, and we are having some struggles right now. So then, for the neural activity, after perfusing the mice, we took their brains and we performed iDisco. So I mentioned before, iDisco is a clearing technique to make the whole brain transparent. And then you coupled it with CFOS immunolabeling, so which is an imaged early gene to detect neural activity. So this is a paper from Davoudian that has already been done. This is my data. You can see that all the little dots are activated cells, which is pretty cool. So these are the results that we had. It's a bit of a mess, and I obtained this yesterday, so sorry, I will not say so much about that. But what is really interesting to say overall is that with these doses, so 150, we really have more brain regions that are activated, much more differences. And if you look a bit about the regions that are activated, you can see that a lot of primary sensory regions are activated and a lot of cortical areas, and we did not see subcortical areas. And normally with CFOS expression, when a region is supposed to be deactivated, you're supposed to see a downregulation of CFOS expressions. So you can also, in a way, attest for a downregulation of the neural activity, but it's much more complicated to target. So finally, the discussion. So our LSD will, our study will be the first one to draw the LSD dose response curve in mice with consistent measure of head-foot response. Then we will better characterize LSD-evoked acute neural activity in whole brain in a dose-dependent manner. Then we will determine what is a low dose and high dose. A low dose meaning being maybe therapeutic, but not having effect on the locomotor activity. And a low dose and high dose may be a dose that affects also the dopaminergic system. So maybe with other actions. So then we want to assess the effect of LSD on the long-term scale to see what it does, because we still don't know, and to focus on the region of interest that we have identified. And what we can do in animals is also that we can manually activate some regions or downregulate some regions, because we can use optogenetics. So then we can have causality, and this is the main thing that is important with animal research. Although our study have a lot of limitation, and CFOS, for example, expression is a good limitation, but also the iDisco technique, because it's a 1 month experiment, and of course in 1 month there's a lot that can happen, a lot of things that can go wrong, we all know that, so there is always limitations. So thank you for your attention.
Thank you, Gabrielle. As before, if you have a question, just raise your hand and someone with a microphone will come to you.
Thanks a lot. So just so that I understand, you have a 30-minute baseline in the open field, then you inject, and then another 30 minutes, then you sacrifice into the iDisco.
Oh, yes.
But I think CFOS takes about 1.5 hours to be expressed. So that way you're actually capturing the cells that were even before the baseline active.
Yeah. So this is a good question, and this is something that has been reported in the literature. Actually, people do not really agree with that. Like, some people say it's 90 minutes, so 1 hour 30, and some people say 30 minutes. And then in between you have a long moment. So I think some CFOs will be activated, and we might miss some others. But this was an arbitrary decision that we took.
And are you considering, like, you're looking at locomotion, just do fiber photometry in, I don't know, dorsal striatum, and look in vivo during the behavior of the mice?
This can be nice. So what we are starting to plan to do next is, because we identified some regions that were overactivated with LSD, we were thinking about using indeed other techniques so we can really capture those regions more precisely when the mice is doing the behavior, or even just activating those regions, like with optogenetics. So this is something that we can do next, but for the moment we just have those results and we still need to analyze them. So
Thanks, and good luck.
thanks.
Hi, thank you for your talk. Could you comment, or do you have any idea what the head twitch response might actually, what could that be in terms of a human response or human experience, especially given the dose response curve, and that it decreases with dose at some point, but there is also a head twitch response there already at a low level at baseline?
Yeah, so it is a bit difficult to find something similar in humans that the head twitch response. And the head twitch response is really a proxy for the hallucinogenic potential, meaning that some authors have done some studies, and if a compound is doing hallucinations in humans, then he will do head twitch response. But now there are some people that are thinking that head twitch response is a sort of tic behavior that is induced by the decreased temperature of the animal, because psychedelics were found to decrease the temperature, and this is also consistent.
And why would it decrease then at the highest doses?
Sorry?
Why would it then decrease again at the highest dose?
Because the temperature follows the same also, those response curve. Those are a bit mystery. I do not have the answer, and those are still questioned by researchers.
Thank you.
I have a question. First, I would like to thank you for the presentation. That was really cool. And second, I would like to ask you a a little bit about your experience or your team experience of getting the approval of using LSD in these studies, given that you come from France, that is not has different regulations than Switzerland. And also one other question, like why do you think that your team used just male rats? I can guess that maybe it's because menstrual cycle in rats, hormones affect, but I think it's really important also to do the research in female rats, because these medicines also will, then in the future will be used by females in general, like humans, and could be important to also know like the other implications.
Mm-hmm. Yeah, I fully agree with you. Thank you for this remark. In animal research, we are using a lot of male mice and rodents because we say that indeed female has hormonal cycle. And then we are just like discarding it because it can perturbate the results. But now we found that hormones are also in males. Wow. And males have also cycles of aggressivity. So at the end, it's a bit wrong to do only male and not female. But for our study, it would just like to make it simple. We already have 7 doses, meaning a lot of animals. So if we wanted to have female plus male, meaning that we have to double the number of animals. So this was the only answer.
Yeah.
Just to be clear with that. And then for the legislation in France, it was a mess.
Yes, but like
yeah, it was really complicated.
Because I come from a university in Italy, so I am interested in these studies, but I think that I just think about the paperwork and just like freeze,
Yeah. So my doctorate student, she waited more than a year before starting her PhD, just because she had to go all over those approval. And in my lab, a doctor, Luc Mallet, is working in humans, and he launched, he's trying to launch a clinical studies using LSD for alcoholism. It's been 3 years that he's going through the administrative process.
Yeah.
It's a lot.
Sorry for just taking the microphone, but what do you think about like using also like derivatives of LSD? Will be like research chemicals like that are legal. Is it possible or?
Yeah, it's possible. I think you have to be sure, like you have to know that it's different from LSD, and then to know that you cannot, if someone is taking LSD and then you're testing another drug that is not LSD per se, indeed the results cannot really be applied to the real life. But then with the derivative also, I wanted to share this new findings also to you, is that lizuride, which is non-hallucinogenic compounds, were found recently to have antidepressant effect in mice. And this is pretty cool for the future, because maybe, we don't know, but maybe the hallucinogenic properties are not necessary. And this is also why we need more research.
Thank you.
Okay. I wanted to ask, maybe you studied something about this as well, or are there any research about animals that have, for instance, PTSD or different disorders? What is the difference between the animals and people? Because I can imagine it's different, but I cannot really grasp it. Could you tell me about it a bit?
So first, I want to be clear with that. When we're talking about we never talk about disease in animal. We talk about model of disease, because we cannot know if the animal is ill. So, for example, for PTSD, it's really hard, I will say, for the animal. Like you really stress the animal really often. all the time. Or you induce him a really strong stress. And so one of the graphs that I showed you, maybe I can go back to this, it was with PTSD model of animal. This one with Ross. So PE and PSS, it's a model of prolonged stress in mice. So this graph, like, it's PTSD mice with big.
And is the research similar to humans? Like, do we really see?
So we really see some similarities. For example, they have some tests. You can put the mice in a box with two parts, separated parts, and in a part you associate it to a stressing stimulus. So really something that is intense. And then you put again the mice and going away in the box, and you will see that he will never go to these places where he has been stressed. He will always avoid it. Yeah. And also there have some experiments with memory. You also have impairment in memories. So there are some tests in mice to assess for the memory, for example, the water maze. So the mice is just like trying to find again the platform, or also a maze. So the mice just learn how to walk in the maze and how to go out.
Yeah, that's really cool. Thank you.
Thank you for your presentation. I was curious about the dose. So you start with 25 micrograms per kilogram of body weight, if I read that correctly. And I was just curious for what was the decision behind that? Because if you compare that to human doses, that is a lot larger, right?
Yeah. So the thing is not really comparable, because mice and humans do not have the same receptors. So there are some differences. And with mice, what have been done in research is that you calculate the dose with the kilograms. So it's always higher, but strangely, I don't exactly know why, the 100, 150, and 200 are also in the mice the therapeutic dose. I don't know exactly why, but it's kind of the similar thing. And those doses exactly, so this is LSD tartrate. And in animal research, previously we were using LSD base, which is a different substance. And LSD tartrate, it has 68% of the active substance in it. And so we just took previous experiments that has done LSD research, so really old research. We looked at the doses that they used, and we converted them to LSD tartrate. So, for example, 500 can seem really, really high, but we have also to keep in mind that it is LSD tartrate.
Thank you.
Maybe one last question?
So in rodent research, facial expression is always or often measured to make a statement about the state of the rodent. So my question is, did you observe any change in facial expression on rats that or mice that got LSD?
In facial expression?
Yeah. So often it's measured like if they are in pain, you measure the facial expression to examine, yeah.
Okay, I see what you mean. So to explain everyone, there are some physical tools that we can use to see if the mice is suffering or not. Namely, if the ears are at the back, the eyes are really closed, the fur is really, then the mice is stressed. We did not see that at all. So this is good. But this is the only thing that we can see, because then it was also difficult to see. You saw also the quality of the video is terrible, so we're not really able to do so.
Okay, thank you very much.
Thank you again, Gabrielle. Very interesting talk, and all the way from here from Paris.